Good morning, children. Today we are going to discuss application of biotechnology. Before reading this lesson, we want to know about DNA, protein synthesis and genetic engineering. These are the basic concepts for biotechnology. Genetic engineering involves in the manipulation of DNA and the production of therapeutically important proteins. It also involves in the extraction of the gene from one organism and transfer to the another organism. That means transfer to the DNA of the another organism of the same species or different species. In this way, producing DNA is known as recombinant DNA. This technique is called recombinant DNA technique. These techniques included in the biotechnology. So what is biotechnology? Biotechnology defined as the application of scientific and engineering principles to the processing of materials by biological agents to produce goods and services. Once again I am saying, so biotechnology defined as the application of scientific and engineering principles to the processing of materials by biological agent to produce products or goods and services. Biotechnology is a umbrella term because it covers all the techniques. Biotechnology is an umbrella term, it covers all the techniques using the properties of living organisms and produce number of the products and provide services. The term biotechnology was first used before 20th century. In the traditional activities as making idli, dosa, bread, wine and curd, etc. Now it is not considered as a biotechnology in the modern sense. In this chapter, we are going to read application of biotechnology in various fields, especially medicine. The recombinant DNA technology was used for prepared number of the hormones in the earliest times. Nowadays, we are using the recombinant DNA technology for producing different type of the hormones and therapeutically use of the proteins. Now we are going to discuss recombinant human insulin. Human insulin is synthesized by beta cells of islets of the Langerhans of pancreas. It is formed by 51 amino acids. It has two chains. One is alpha chain, another one is beta chain. These two chains are held together by the disulfide bond. A chain, that means alpha chain, contain 21 amino acids. Beta chain, that means B chain, contain 30 amino acids. The insulin is very important to maintaining the glucose level in the blood. Whenever its amount is decreasing in the body, it leads to the diabetes mellitus. If untreated, this condition leads to the death. So, insulin is very important to control the blood glucose level. Usually, insulin converting the blood glucose and glucose is converted into glycogen and stored in the liver. Whenever this amount is decreasing in the blood, it leads to the diabetes mellitus. That is controlled or treated by the help of insulin. The first best and bad thing in 1921 isolated insulin from the islets of the Langerhans of dogs. And they demonstrated and its effectiveness against for the diabetic person. In the earlier years, the insulin was isolated from the cows and pigs. That means islets of the Langerhans of the cows and pigs. This insulin given to the diabetic patient in the early years. But there is some differences in there, that means minor differences in the structure of the human insulin and animal insulin. For this created some critical allergical reaction in the diabetic patient body. The recombinant technology was started in late 1970s. In this technique, they used the DNA as a vector. That means E. coli DNA, that means bacterial DNA is as a vector. In this technique, the small gene is isolated or extracted from the human insulin. Then it is transferred to the plasmid of the E. coli. So they selected the one bacterial cell. From that bacterial cell, they removed the plasmid. By the help of the restriction enzyme, they cutted the small piece. In that place, this gene is replaced with the help of the ligase enzyme. Then this recombinant plasmid is introduced into the another one bacterium. That bacterium is known as recombinant bacterium. Then these plasmid multiply or replicated number of the time and produce different kinds of the polypeptide chains. These polypeptide chains are multiplied with the help of the fermentation tank. Then it is removed from the fermentation tank and arranged in the ladder-like structure. First produced insulin was pre-proinsulin. This pre-proinsulin consists of two segments. One is A segment, another one is B segment. These both segments are connected by the C chain. Then this pre-proinsulin is translated into the another one structure, that is called the proinsulin. This pre-proinsulin only is the precursor of the insulin. Then the proinsulin existed the C chain and formed the pure insulin. This pure insulin has a two chain, A chain and B chain. In this way, insulin produced by the recombinant DNA technique. These are used for the treated the disease or disorder of the diabetic person. Insulin was the first products produced by the recombinant DNA technology. In 1982, it was approved by the government to use insulin for treatment of diabetes. In 1986, it was marketed under the trade name Humulin. So next, we are seeing about interferons. How the interferons are produced by the DNA recombinant technique. What is interferon? Interferons are the proteinaceous substance and antiviral substance. These are produced in the mammalian cell when it is affected by the virus. These interferons are made up of, or these interferons are discovered by Alec Isaac and Jean Linderman in 1957. These interferons classified based on the structure into three types: alpha interferon, beta interferon and gamma interferons. These interferons are isolated from the blood, but amount of blood required for this isolation of the interferon is enormous, but not practical. So to overcome this issue, interferons are produced by the recombinant DNA technique. The yeast Saccharomyces cerevisiae is used for protection of recombinant interferons. These interferons are produced by the help of yeast than the E. coli. The yeast Saccharomyces cerevisiae was used for preparation of recombinant interferons than the E. coli, because E. coli does not possess the machinery for glycosylation of proteins. These interferons are used for treated diseases like AIDS, multiple sclerosis, cancer, hepatitis C, and herpes zoster. These interferons could not reach to the common man because its preparation cost is very high. So next we are going to see how the other substances like hormones and blood clotting factors are produced by the recombinant DNA technology. But this hormones production and the blood clotting process are not have in our book. But for the knowledge we want to know about how the hormones are produced by the recombinant DNA technique. We know already the growth hormone is very essential for the growth of the body. Somatostatin, somatotrophin, these are the growth hormones. These growth hormones are produced in the pituitary gland. So these growth hormones are giving or using as a life-saving drugs. Whenever the growth hormone amount is decreasing in the body, it leads to the dwarfism. So avoiding this disorder, we want to use synthetically growth hormones. These growth hormones are produced by the DNA recombinant technique. So in this technique, they are extracting the gene from the pituitary gland and that is attached or transferred to the E. coli of the bacteria cell. So with the help of the same method what we are already followed in the insulin production, like that here also the plasmid is used as a vector and the gene is attached to the or transferred to the plasmid, and this plasmid is introduced into another one bacterium. The bacterium is used like a recombinant bacteria. From that bacteria, that is amino acids are extracted and produced the growth hormone. In this way we can produce another one substance, that is a blood clotting factor. There is a 12 blood clotting factors are there. But the 8th blood clotting factors or blood clotting process, we are making the 12 steps. The 8th factor is very important for the blood clotting. This factor is present in the blood. So it is produced by the recombinant DNA technique. The gene is extracted from the blood. The gene is present in the X chromosome. The 8th factor is present in the X chromosome. That gene is extracting from the X chromosome and transmit to the E. coli. Maximum we are using the bacterial cell for the recombination technology. So that is transferring to the E. coli and producing the 8th factor. This 8th factor, that means a blood clotting factor, is essential for giving the treatment for the hemophilia disease. Like this, so number of the other products, that means medical products, are produced by the recombinant DNA technique. We can say the vaccine and a different type of the medicines like that. Thank you.